Okay, so alright. So I didn't manage to get to this PowerPoint yet, even though the exam is tomorrow. So I figured it'd be better to go through it than to just not go through it. So the first thing is the PIP2 pathway. And the PIP2 pathway is basically how you can link together, microbial detection and uptake with outcomes like phagocytosis and reactive oxygen species production and stuff like that. And it's basically just a signaling pathway. So the PIP2 pathway you should associate with G protein coupled receptors. Any of the pattern recognition receptors that are G protein coupled receptors can initiate the PIP2 pathway. And they're going to bind to their ligand, usually a PAMP or an antibody or complement protein, and trigger this pathway. So the G protein coupled receptors in the innate immune system include the FMLF receptor, the C5a receptor, and probably the C3a receptor as well, antibody FC receptors, and complement receptors, CR1 through 5. So those are your GPCRs. The way that it works is that normally in a resting state, G alpha is bound to GDP, and it's also bound to G beta and G gamma. So it's a trimeric G protein composed of 3 subunits that are non-covalently bound. And G alpha is bound to GDP. None of the proteins in the trimer are associated with the receptor initially. Okay. However, when the receptor binds to its ligand, depending on what receptor it is, it'll have a different ligand. That will induce the G alpha beta gamma trimer to bind to the intracellular receptor tail of the receptor, and then the G alpha protein releases the GDP molecule that it's bound to, and it binds to a fresh molecule of GTP. Okay, so it doesn't phosphorylate the GDP; it actually releases it and picks up a new one. And most of the this nucleotide is in the form of GTP in the cell. So this causes these proteins to dissociate from the receptor. So they're only associated for a short time while GDP is released, and then GTP binds. That causes them to dissociate. G alpha dissociates from G beta gamma. So now you really have 2 G proteins dissociated from one another. And G alpha and G beta gamma activate distinct proteins. Okay. The end of the pathway, like I said, results in things that you need to see in response to microbial recognition. Chemotaxis, reactive oxygen species production, increased microbicidal capacity, synthesis in some cases of pro-inflammatory cytokines. It also influences metabolism. It switches them to mostly glycolytic metabolism as opposed to, fatty acid burning cells. They switch to glucose burning cells. It influences gene expression. So cytokine genes can be made. Genes that encode ribosomes can be made. And cell division will also occur a lot of the time. Because that macrophage that recognized that pathogen is probably going to need reinforcements. So why not replicate? So G proteins look like this. Their N terminus is outside the cell. Their C terminus is inside the cell. And they are 7-pass transmembrane proteins. And they have this sort of structure to them. And this is a conserved structure of a G protein coupled receptor. So this is a GPCR. And G alpha and G beta gamma, they are linked to the membrane by farnesyl groups. And this is a post-translational modification that is done to these proteins by an enzyme in the cytoplasm. This here is the figure that Sanders uses for the PIP2 pathway, and I absolutely despise it because it's really blurry and really kind of confusing. So I made my own, and my own looks, more like a wall of text, but it's a lot clearer. So G alpha is bound to GDP, and it's bound to G beta gamma when it's inactive, right? Then the G protein coupled receptor binds to its ligand and it gets activated. This causes G alpha to release GDP, and to bind to a fresh molecule of GTP. Now you have G-alpha GTP. This is the active form, and it's still linked into the membrane, right? You also have that dissociating from G-beta gamma, which goes off and does its own thing. And G-beta is also farnesyl linked. Okay. So these are two distinct pathways. The G-beta gamma pathway is a little simpler. So we'll start there. So G-beta gamma. What happens here is G-beta gamma activates a protein called PREX1, which is a guanine exchange factor for a small G protein in the cytoplasm known as RAC2. So PREX1 becomes activated by G-beta gamma, and it goes to RAC2 and it says, hey RAC2, release your GDP and bind to a new molecule of GTP. So RAC2, what does it do? It releases GDP and it binds GTP, and then it is activated. And what it does is it takes some of the components of NADPH oxidase, which is an endosomal enzyme, and it assembles the complete NADPH oxidase enzyme in the endosome membrane. Usually 3 of the 5 subunits of NADPH oxidase are in the endosome, and 2 of the 5 are in the cytoplasm. And when they're separated, this enzyme has no activity. However, when RAC2 swings by and it brings them all together as one big happy family, now NADPH oxidase is assembled, it is activated in the endosome membrane, and it pumps the endosome full of reactive oxygen species. And the microbe is going to be shredded in the endosome by these reactive oxygen species. Okay, so that's the G-beta gamma pathway. It results in reactive oxygen species generation by NADPH oxidase. The G-alpha pathway is a little bit more complicated. It only activates one protein, and that protein is phospholipase C. Phospholipase C. What phospholipase C does is it cleaves a molecule called PIP2 in the membrane, and it cleaves it into 2 components. It cleaves it into a hydrophobic diacylglycerol, DAG component, and a hydrophilic inositol triphosphate molecule, IP3. And that is kind of like a sugar molecule with 3 phosphates attached to it. So it kind of cleaves off the head group of PIP2, and that results in IP3 and DAG. IP3, inositol triphosphate, it diffuses through the cytoplasm and goes to the endoplasmic reticulum. On the endoplasmic reticulum, there is a transmembrane protein that serves as a receptor for IP3, and it's simply called the IP3 receptor, IP3R. When IP3 binds to the IP3 receptor on the ER, it triggers IP3 receptor to open. And the IP3 receptor is actually a calcium channel. It's a ligand-gated calcium channel, and that triggers calcium ions to be released from storage in the ER. Normally, calcium ions are sequestered or concentrated or stored in the endoplasmic reticulum, but when IP3 binds to its receptor, it triggers the calcium ions to be leaked out of the ER and into the plasm. Now we have calcium ions leaking into the cytoplasm. We've got an influx of calcium. Those calcium ions bind to and activate a protein called calmodulin. Cal for calcium, because it binds to calcium. Two ions of calcium are going to bind to calmodulin, and calmodulin turns on proteins that cause adhesion and chemotaxis by rearranging the cytoskeleton. So it's going to cause actin to depolymerize and then repolymerize in the direction of the stimulus. So for example, if you stimulate the cell with C5a and most of the C5a is left of the cell, then the cell is going to move left. Okay? And the C5a, as you probably remember, C5a is a chemotaxin or chemotactic factor that directs movement toward the source of the problem. So the cell is going to move in the direction of the C5a that's being produced from the microbe. Okay? It's going to be moving toward the source of the problem. So the C5a receptor is one thing that can trigger all this, and it's going to move in the direction of the C5a concentration gradient. It's going to move toward higher concentrations of C5a. PIP2 is cleaved by phospholipase C into IP3, which does all that, but it's also cleaved into diacylglycerol, DAG. DAG, this is still a hydrophobic molecule, so it's still in the membrane, and it stays in the membrane. It has two things that it does. First of all, it activates protein kinase C, PKC. And PKC generates reactive oxygen species and or induces degranulation of the cell by mobilizing granules. Okay. In the case of macrophages, you don't really have degranulation. You have what is called frustrated phagocytosis. And this is when something is too large to be taken up by a phagocyte, for example, a large yeast cell or a large parasitic worm. Those things are too large to kill inside of the macrophage because the macrophage isn't as large as them. Instead, the macrophage is going to degranulate and release reactive oxygen species and other antimicrobial substances in the direction of the pathogen, and it's going to kill the pathogen. Unfortunately, frustrated phagocytosis comes with a little bit of collateral damage, generally speaking. Not all of those reactive oxygen species are only going to act on the target. They may accidentally act on tissue cells that are near the target, and they can also diffuse and kind of end up in anyone's guess where they're going to go. However, they're pretty short act. They're going to be neutralized relatively quickly, and cells have ways of dealing with oxidative stress, so the collateral damage is going to be minimal at best, but you can accidentally damage host tissues with frustrated phagocytosis. Okay, so that's protein kinase C. It results in those things, and the way that it brings about these things is by phosphorylating target proteins in the cytoplasm. The exact target proteins that it phosphorylates are outside of our scope, and it also depends on what cell you're working with, if it's a macrophage versus a mast cell, and also the subset of the cell. There's different kinds of macrophages. There's different kinds of mast cells. They're going to have different target proteins inside the cytoplasm. But the effect is going to be tailored to what the cell needs to do, right? If it's a large target, it's going to undergo frustrated phagocytosis. If it's a mast cell, it'll undergo degranulation. And if it is a macrophage with a relatively small target, like a bacterium, then it's only going to make reactive oxygen species. It's not going to do frustrated phagocytosis, okay? So that's protein kinase C. Diacylglycerol not only activates protein kinase C, it also activates phospholipase A2, PLA2. And this is an enzyme that cleaves a molecule called arachidonic acid from membrane lipids. Arachidonic acid is a fatty acid with 20 carbons. Okay, 20 carbon chain with a fatty acid on one end, a carboxylic acid group on one end. And arachidonic acid is converted into this is, when this cleavage happens, it results in two ultimate products. It results in a group of compounds called the eicosanoids, which are arachidonic acid derivatives. So arachidonic acid undergoes catalytic conversion into, a bunch of different eicosanoids, depending on the cell type. Different eicosanoids are going to be made depending on the cell type. And platelet activating factor is also going to result. Okay, so part of the molecule is turned into eicosanoids. The rest of the membrane lipid is turned into platelet activating factor, and that is true for all cells that produce eicosanoids. They're going to make platelet activating factor. The exact eicosanoids you do not need to know. You probably also don't even need to really know about the structure of platelet activating factor or anything like that. All that you need to know about phospholipase A2 is that it cleaves arachidonic acid from the membrane, and that results in eicosanoids, and it also results in platelet activating factor. Okay, and it's triggered by DAG. So that is the PIP2 pathway. That is everything that you need to know about it. There are a lot more specific details, and you could probably spend an entire PhD studying the PIP2 pathway, but everything that you need to know about it was right there. So, there's some points of confusion, probably, already. There's a couple of different phospholipases. You've got phospholipase C that's activated by G-alpha. You've also got phospholipase A2 that's activated by DAG. Phospholipase C, it cleaves right here. And it generates IP3 and DAG from that compound, from this membrane lipid, this generic membrane lipid. On the other hand, phospholipase A2, it cleaves the fatty acid off of carbon 2 of the glycerol backbone of the fatty acid, of the phospholipid. Okay, so that results in arachidonic acid being released, and then the rest of the molecule will be turned into platelet activating factor through a couple of steps of, enzymatic catalysis. All right, so this is a figure from Pathologic Basis of Disease. I would not consider this to be very important material, but this just shows you all of the different eicosanoids. It's very complicated. So you can see that phospholipase A2 is going to cleave arachidonic acid from the cell membrane phospholipids, and then depending on the cell type, arachidonic acid is catalytically changed into all of these different eicosanoid molecules. And there's just like literally a whole laundry list of them. And it depends on what cell type you're working with, the pathway that's going to be invoked. I actually tried to learn this pathway one time, and yeah, no one really has ever tried to piece this all together. So don't worry about knowing any of the eicosanoids, in my opinion. In general, though, what the eicosanoids do, basically they are inflammatory mediators. Some of them result in different things than others. So you've got some that cause vasodilation, some that cause vasoconstriction. Those that cause vasoconstriction are important in things like asthma. You're looking at constriction of bronchioles in the lungs. Some of them cause increased vascular permeability. Some of them cause chemotaxis, and they upregulate leukocyte adhesion. You don't need to know which of these do which. Just know that they're kind of generally pro-inflammatory, and that there's a bunch of them. The major groups are the leukotrienes, the prostaglandins. Those are the two major ones. You've also got the lipoxins, the heats, and thromboxane, which is made by platelets. And then the other portion of the membrane phospholipid after cleavage, so part of it is turned into arachidonic acid. The remainder of it, so the stuff that's still attached to carbon 1 and carbon 3, as well as the glycerol backbone, it's all turned into platelet activating factor. And the major sources of that are pretty much everything that makes everything you see down here, as well as endothelial cells. And platelet activating factor is just a very potent inflammatory molecule. It's going to activate immune cells, and it's going to cause vascular permeability increases and vasodilation. So it's inflammatory and it makes cells angry. Okay. This pathway here, not really within our scope though. But I figure familiarizing you with some of the names of these things may be beneficial to follow what's happening in class. Perhaps a bit too late for that though. This slide here, you can put it out of your mind. You don't need to worry about the eicosanoids. And then this is a table sort of simplifying them, but again, you do not need to know them. So this one here shows you a couple of cool things. So this is when a neutrophil eats something up, like a pathogen, right? So it takes it up through sometimes always a pattern recognition receptor. Sometimes a G protein coupled receptor is involved. For example, the FMLF receptor is going to recognize bacterial polypeptides, and it's going to cause RAC2 to become activated by means of PREX1, excuse me, and RAC2 is going to assemble NADPH oxidase in the endosome membrane, and that forms the superoxide anion. Superoxide anion is converted by way of enzymes into a bunch of other reactive oxygen species like hydrogen peroxide, hypochlorite, etc., by different enzymes. And also you're going to have the lysosome fusing with the phagosome. You're going to have secondary and primary granules fusing with the phagosome. All of these things are storing different toxic components like enzymes in primary granules. NADPH oxidase is actually localized to secondary granules, but it's all going to be fused into one big angry endosome that eventually fuses with the lysosome. So that bacterium is going to be shredded by more ways than you can probably count on both hands. There's no real way for them to avoid all of these different mechanisms. So there you have it. And then this is another figure showing you basically the same thing, except this one zooms in a little bit better on the NADPH oxidase. You're going to see this figure later when we talk about exam 3 material. NADPH oxidase takes oxygen and turns it into the superoxide anion O2 with a single lone electron, and it has a negative charge. That is converted into hydrogen peroxide by myeloperoxidase, and that is converted into, or excuse me, that occurs by way of superoxide dismutase, and then that's converted into hypochlorite by way of myeloperoxidase. And hydrogen peroxide also naturally decomposes into the hydroxyl radical, which is toxic as well. So you're producing a number of different reactive oxygen species by first synthesizing this and then converting it into other stuff. And right here you see the iron +2, ferric iron. Ferric iron can catalyze the conversion of peroxide into the hydroxyl radical. So it can diversify the types of reactive oxygen species that are being produced. This is called the Fenton reaction. You might remember that from micro if you took micro. This is the Fenton reaction. And then you also have this enzyme called inducible nitric oxide synthase that is expressed by innate immune cells, your phagocytes and your eosinophils. They take arginine and they turn it into nitric oxide. And nitric oxide is also toxic in high levels, which it will be in high levels in the endosome. It's toxic to basically all forms of life, including bacteria, fungi, parasites, etc. So you also have that going on. So you call these reactive nitrogen species, right? Because they contain nitrogen. Altogether, we refer to these toxic components as reactive oxygen and nitrogen species, RONS. Okay. Most of them are oxygen-based, though. Only nitric oxide and another compound are nitrogen. So this is the pathway. It's showing you the actual chemistry that happens. You can see we start with NADPH oxidase, taking oxygen and turning it into superoxide anion. That is converted into peroxide by superoxide dismutase. Peroxide has a couple paths that it can take. It can be converted into hypochlorite, which is basically bleach, and there is by way of myeloperoxidase. That's an enzyme you're going to want to know. It can also be converted into the hydroxyl radical by iron. This is the Fenton reaction. And some pathogens express the enzyme catalase to break down hydrogen peroxide into the non-toxic water and oxygen molecules. So catalase is a way of avoiding reactive oxygen species, and it's expressed on some pathogens, especially aerobic pathogens. Superoxide anion can combine with nitric oxide, which is formed by nitric oxide synthase, and it can combine with nitric oxide. Nitric oxide by itself is toxic, but it can combine. These two can combine and form another toxic compound called peroxynitrite, and that is toxic as well. It's just another free radical. All of these things in summation are going to kill the pathogen. They're also going to turn on certain enzymes in the endosome that induce that induce the killing of the pathogen. It's going to turn on certain antimicrobial enzymes. It's also going to allow for potassium ions to enter the endosome, and potassium ions are important for the generation of the acidic pH in the endosome. So potassium ions, they turn on proton pumps. Proton pumps acidify the endosome, and most of the toxic enzymes in the endosome are activated at low pHs, like 4 or 4.5, maybe 3.5. That kind of range is where they're turned on. And at that point, they shred the pathogen. So reactive oxygen species go through this sort of pathway here. Host cells have the enzyme glutathione peroxidase, which converts peroxide into water. Catalase is expressed on certain bacteria and other pathogens that can break down peroxide. So we have ways of removing free radicals, and some pathogens can also sort of avert this by expressing catalase that detoxifies free radicals. Iron can convert peroxide into the hydroxyl radical, which is not so safe and cannot be broken down. And it's also much longer lasting because there's nothing that can break it down in the cell. And the hydroxyl radical is probably the most toxic of all of these because it's the longest lasting. It can diffuse to the membrane where it peroxidates lipids and causes membrane damage. You can open holes in the membrane. It can oxidize proteins and cause them to misfold and denature it down. It can also induce them to literally split apart into pieces. And regardless, you're going to have a malfunctional protein anyway. And it can also diffuse to the nucleus, or it can diffuse to the DNA in bacteria, which is not in a nucleus, and induce mutations and single and double-stranded breakages in the DNA backbone and cause mutations that way. This is one of the reasons that chronic inflammation over many years can result in cancer, is because the reactive oxygen species produced by macrophages, if you get enough of them and if they're delivered over a long enough time period, you can have enough reactive oxygen species produced to where you get a significant amount of mutation to where it becomes likely that you're gonna have proto-oncogenes converting into oncogenes, and you're going to have mutations accumulating in tumor suppressor genes. And this is why some pathogens that induce a chronic inflammatory response are associated with cancer. This is not the only way that infections can cause cancer, but it's one of them. One example is Schistosoma hematobum, which is a worm that produces eggs that are transported into your bladder. And those eggs induce a strong inflammatory response, but the worms are not killed. They continue producing eggs. So you get a bunch of eggs over a long time period, inducing a lot of inflammation and reactive oxygen species production. And that results in mutation to some of the bladder cells, so it can result in bladder cancer. Just one of the ways that inflammation can induce cancer. So yeah. So some more things that reactive oxygen species and nitrogen species can do. They're listed here. You can halogenate some of your lipids and proteins and DNA by hypochlorite and hypobromite. These things can attach bromine atoms and chlorine atoms to different macromolecules, and that disrupts the functioning of those macromolecules, and so they don't work anymore. They're going to have to be replaced with fresh ones. They also do all the other things that are listed here. So this is a figure you're now seeing for the third time, but now you can understand some of these toxic oxygen and nitrogen species right here a little bit better. And then this is a figure that I think you're seeing for the second time. It just shows you where neutrophils store some of these things. They have azurophil granules, which are primary granules. They're a little bit larger. And then they have secondary granules or specific granules, and they're a little bit smaller. They contain different things than the primary granule. But anyway, they store a lot of stuff separately so that you don't get the activation of certain things when it shouldn't be activated. For example, elastase is stored, I believe, in secondary granules and cleaves and activates the beta defensins and turns them on, which can accidentally be toxic to the cell, but is something that we're willing to accept when there's a pathogen inside the cell. We want to be making beta defensins that are active. So the fusion of the primary and secondary granules results in the cleavage of beta defensins into the active form, and that can kill the ingested pathogen. So then we have adhesion molecules, and these are definitely something that you want to study in good detail. There's the selectins, integrins, and then the immunoglobulin-like superfamily of proteins. So there's 3 families of proteins. The selectins, they are weakly adhering molecules. They're the first step in extravasation. They are transmembrane glycoproteins expressed on activated endothelial cells, and they promote rolling adhesion by leukocytes. Selectins have a distal lectin-like domain, and that's their binding domain. It binds to fucosylated oligosaccharide ligands on leukocytes. For example, the sialyl Lewis X blood group on neutrophils and T cells, and basophils is a ligand for P and E selectin. Then we have the integrins. These are heterodimers. They are transmembrane. They contain an alpha chain and a beta chain linked together by disulfide bond and non-covalent interactions. They're expressed on leukocytes. Their job is to enable tight binding. And what they do is they bind to ICAMs that are expressed on endothelial cells and they also bind to extracellular matrix proteins. And we'll go over some of those ligands. Third, you have the immunoglobulin-like superfamily proteins. They contain immunoglobulin domains that you will also see in antibodies. Immunoglobulin domains are also seen in MHC, and they're seen in the T cell receptor. So immunoglobulin domains are widely sort of conserved. The ICAMs are the intercellular adhesion molecules. That's what ICAM stands for. They're expressed on endothelial cells, and they are in this family. They allow for tight binding. They bind to integrins. So their ligands are integrins. And then you have PCAM, or CD31, and it's called PCAM sometimes. It's called CD31 some other times. You're going to want to know both names for this one. It stands for platelet and endothelial cell adhesion molecule. Okay. It's another member of this family. It's responsible for extravasation, which is basically the very last step during adhesion. This is when the cell physically pulls itself between endothelial cells and into the tissue. Okay. So it's expressed by activated leukocytes and endothelial cell-to-cell junctions. And it's a ligand for itself. So PCAM on neutrophils is going to bind to PCAM on vascular endothelial cells, and that's an interaction with the same molecule just on 2 different cells, and that's going to allow it to pull the neutrophil through the endothelium and into the tissue. And then we've got some nice figures for this. You're going to want to know more detail than that, though. That's just the 3 families. The important selectins are P and E selectin, and they bind to the sulfated sialyl Lewis X blood group on neutrophils, monocytes, and T cells. Tumor necrosis factor causes membrane fusion of these granules inside endothelial cells called Weibel pallade bodies. Weibel pallade bodies are pre-synthesized inside endothelial cells that are waiting to be stimulated with inflammatory cytokines. Weibel pallade bodies store the protein P-selectin. It's pre-made. P-selectin is pre-made and it comes first. And tumor necrosis factor is going to cause the fusion of these granules with the membrane on the blood side of the endothelial cell. Tumor necrosis factor and other inflammatory stimuli will also cause endothelial cells to begin making E-selectin de novo, meaning synthesizing them from scratch. You're going to need to go through transcription and translation and secretion in order for E-selectin to show up. So P-selectin is pre-made, it shows up first. E-selectin is synthesized second. and it's synthesized from scratch. It's not pre-stored. We've also got L-selectin. L-selectin is not important. The integrins that you're going to want to know. LFA1 and CR3 are the two important ones. There's also VLA4 and VLA5. LFA stands for leukocyte functional antigen, so you can imagine that this one is expressed on leukocytes, right? Neutrophils, monocytes, T cells. VLA, very late antigen. And complement receptor 3 is actually an integrin. Not only is it a receptor for complement, but it's also an adhesion molecule. So LFA1, it binds to both ICAM1 and ICAM2. ICAM is for intercellular adhesion molecule. So this one must be expressed on cells that are closely crowded, i.e. it is expressed on the endothelium, hence intercellular adhesion molecule. ICAMs are expressed on the endothelium. CR3 serves as an adhesion molecule by binding to ICAM1, but remember it's also a receptor for IC3b, and it's also a receptor for fibrinogen in the extracellular matrix, and that can allow for some adhesion activity, and coagulation factor X, as long as it's been activated. LFA1 and CR3 both bind to ICAM1, but LFA1 also binds ICAM2, and these two molecules bind to different portions on ICAM1, okay, different areas. Now, IL-8 is not only important as a chemokine, but it's also important as an upregulator of adhesion molecules. It binds to its receptor, CXCR1 and CXCR2 are the receptors for it, on neutrophils, and that causes the activation of a protein called talin. Talin's job is to tightly link LFA1 and CR3 to the cytoskeleton of the cell by tethering them to their shared beta chain cytoplasmic tail. So both LFA1 and CR3 express the same beta chain, but they have a different alpha chain. Remember, these are alpha-beta heterodimers. The beta chain can be tethered all the way through the membrane and into the cytoskeleton by talin. Talin covalently links together LFA1 and CR3 to actin in the cytoskeleton, meaning it strongly links it. VLA4, less important than CR3 and LFA1, but it's expressed on monocytes and T cells, and it binds to VCAM1 on the activated endothelium. So that's one way that monocytes and T cells can leave the blood and enter the tissue. And then VLA5 on monocytes and macrophages, it binds to fibronectin to enable migration through tissues. So once it leaves the blood and enters the tissue, it has to migrate through the tissue. And you can picture them sort of pulling themselves through the jungle using their pseudopodia, which works kind of like two hands. And they pull themselves on fibronectin molecules using VLA5. CR3 is also going to enable this. The immunoglobulin superfamily, we've got ICAMs and other IGSF proteins. ICAM1, it binds to LFA1 and CR3. ICAM2 only binds to LFA1. Now, ICAM2 is expressed all the time constitutively on the endothelium at low levels. This is probably where we get some of our monocytes from, and this is where some of the macrophage replenishment is from throughout your life, even when there's no inflammation. ICAM1 only becomes expressed on the activated endothelium. So if you stimulate endothelial cells with tumor necrosis factor, complement A components, or platelet activating factor, ICAM1 becomes expressed, and it binds to LFA1 and CR3. Excuse me. VCAM1 is another immunoglobulin-like protein. It becomes expressed only on activated endothelial cells, and its ligand is VLA4 on monocytes and T cells. And then lastly, we have PCAM, or CD31. Know both names. It's another member of this family, and it enables the very last step of leukocyte leaving, which is extravasation. This is when the leukocyte pulls itself across the endothelial barrier and toward the basement membrane. Diapedesis is when the leukocyte breaks down the basement membrane to allow itself to enter. PCAM is expressed on activated leukocytes, and it's expressed on endothelial cells at cell-to-cell junctions. So here's a table showing you all of that good stuff. I didn't really like the Janeway table because it didn't show everything, so I made my own. And you've seen this figure plenty of times by now. It just shows you sort of what you should be picturing. These blood cells are going to be leaving between the gaps that are located between your endothelial cells. These are desmosomes. So normally they're linked together pretty closely, but during inflammation, you have retraction of the endothelial cells. They're going to contract and become sort of narrower or less long. And then blood cells can leave and squeeze between them. Okay. So stage 1 is rolling adhesion. Macrophages might produce tumor necrosis factor that acts on endothelial cells. You can also have C5a, histamine, lipopolysaccharide of gram-negative bacteria, leukotriene B4, etc. Inflammatory mediators activating endothelial cells, and that causes vascular endothelial cells to contract, spread apart. They release preformed Weibel pallade bodies containing P-selectin. They synthesize E-selectin de novo. They make it from scratch from mRNA. And they begin to make ICAM1 that's only expressed on the inflamed endothelium. And that allows for tight binding, which is stage 2. So here's what it looks like. Sulfated sialo-Lewis X blood groups on neutrophils are going to bind to E-selectin, and that's when you have rolling adhesion. Remember, the blood flow is going to slow down because of vasodilation, so these are going to be moving relatively slow already. That allows them to interact more intimately with selectin molecules on the vascular endothelium. They're going to start rolling slowly along the vascular endothelium like this. Once they're rolling slowly, they're going to bind with their LFA1 and their complement receptor 3 proteins to ICAM 1 and 2 on the endothelium. ICAM 1 is probably a little bit more of a higher affinity interaction. ICAM 1 allows tight binding. So they stop rolling, they freeze in place. Okay. IL-8 acts on its receptors, CXCR1 or CXCR2, and that causes LFA1 and CR3 to be tightly linked to the actin cytoskeleton by the beta chain. That allows this to stop wobbling on the membrane and instead be stone hard, rock hard. It's linked to the membrane firmly, and the leukocyte stops rolling, it freezes in place. Next, what happens is PCAM, or CD31, on the neutrophil and on the vascular endothelial cell at the cell-cell junction binds together. PCAM binds to PCAM. PCAM on neutrophil binds to PCAM on the endothelium, and the neutrophil pulls itself out. And next you have diapedesis. Okay, so that's extravasation. Diapedesis is when the neutrophil or leukocyte breaks down the basement membrane. It needs to pass through the basement membrane in order to get to the tissue, because that's a semi-solid barrier, right? Now it's in the tissue, it has to migrate using the IL-8 or C5a, C3a, leukotriene B4, whatever gradient. It's going to move toward an increasing concentration of these things so that it can move in the correct direction. So migration is going to happen fourth. And this picture here shows you the interactions between ICAM proteins and their integrin ligands on neutrophils and other white blood cells. I guess read this slide on your own time. I've kind of already explained all this. And then this here was a summary of how extravasation of neutrophils occurs. So if you want to keep track of these steps in an orderly way, this slide welcomes you. So we have less time than I thought we'd have. So my question that I'm posing to you is, do you want to go through the exam cram slideshow, which is like the high yield slides, or do you want to go through the practice exam, which is probably a lot harder and will take more than the time we have? I guess both of them are going to take more than the time we have, but which one would you rather have me do? Exam cram slides or practice exam?
We have access to the high cram slides, right?
Yes.
Okay.
They're on Blackboard.
You
also have access to the practice exam answers. They're on Blackboard as well.
Okay. I'd probably rather go through the exam.
Yeah, I would agree with you. Is everyone okay with that?
Yeah.
Yeah. Okay. Let's see. So there's no applied immunology, and that's good because this exam came so fast. It hit me like a baseball. Okay. So there's still a lot of questions on this regardless, so it's definitely going to take more than the time we have. The first question is, which of the following is a professional antigen presenting cell? B cells, neutrophils, monocytes, or A and C, or all the above, none of the above. So neutrophils are the only one that is not a professional antigen presenting cell. All of the other ones express MHC2 and B7 and can present antigen to T helper cells. Which is true for a professional antigen presenting cell? It can provide secondary activation signals to a Th2 cell. It processes polysaccharide antigens and presents them to CD4 positive T cells and includes macrophages. So this first one is definitely true. Remember, B7 is one of the things that they have to express, and that provides the secondary activation signal. Second one is not true. Polysaccharides are not presented in MHC2. Okay. Polysaccharides really aren't presenting to T cells at all. But macrophages are definitely an example of a professional antigen presenting cells. This one's A and C. Okay. Number 3, which is true for macrophages? A, it can produce IL-2, B, interferon gamma, C, IL-12. Only C is true for this one. The only sources of IL-2 are T cells, and that's about it. ILC1s might be capable of it. I'm not 100% sure. IL-2 only comes from T cells. Interferon gamma, the only sources of that are natural killer cells and Th1 cells. Probably ILC1s as well, but we don't care as much about those. Which of the following may cleave C3? So there's a lot of things that can cleave C3. C3b B flat, that is the alternative pathway C3 convertase, so yes. Factor I does not cleave C, or I guess technically it cleaves C3b, but it doesn't cleave C3. So factor I is not a correct answer here. It doesn't cleave C3 itself. It breaks down C3b. C4b2b 3b. That is the classical lectin pathway C5 convertase. So that's not an answer here. C1s, that is the enzyme that cleaves C4 and C2 in the classical pathway. So nope. MASP1, this one's actually true. MASP1, remember, is a promiscuous protease, and it can cleave molecules of C3 to some extent, even when it's not activated. So MASP1 can cleave C3. Factor D, nope. Fixed IgA can cause the cleavage of C3. This is one way that the alternative pathway can be activated. C-reactive protein, nope. Okay, which is true for elimination of intracellular pathogens? It requires a Th1 response if not eliminated by innate responses. That's true. Th1 cells are going to be required for killing intracellular bugs. Natural killer cells may function in the context of innate and adaptive responses. That one is true. Natural killer cells are going to be activated by Th1 cytokines like IL-2, which is going to cause them to grow and proliferate, and tumor necrosis factor. Tumor necrosis factor is going to upregulate natural killer cell activity. And also IgG produced by adaptive immune cells can activate natural killer cells as well. So they are functioning under innate system and adaptive system. They're enhanced by the adaptive system. We consider them innate immune cells though. Interferon gamma can improve macrophage killing of ingested pathogens. That one is certainly true. It makes them better killers. Interferon gamma comes from natural killer cells and Th1 cells. What cytokines are made by Th1 cells? This is going to be interferon gamma, tumor necrosis factor, and IL-2. Those are the three to remember. Which is true for the adaptive immune response? Multiple answer. Excuse me. CD8 positive T cells, or CTLs, require T cell help for a secondary signal to kill virally infected cells. This is not true. CD8 cells don't require B7, and they don't require cytokines in order to go into a tissue and kill a virally infected cell. They already received B7 in the lymph node from dendritic cells, so that one's not true. Naive B2 cells require T cell help to proliferate and differentiate. That one is generally true. Notice that says B2 cells, so these are not your weird B1 cells. These are your classical B2 cells. Yep, to proliferate and differentiate, they require T cell help, generally speaking. Primary signal for activation of a Th1 cell is interferon gamma. Not true. Primary signal is always the T cell receptor binding to MHC2. Activation of naive CD8 cells can be enhanced by Th1 cells. That is absolutely 110% true. IL-2 and interferon gamma can both serve as a secondary signal for activation of CTLs when they're naive. Only when they're naive. When they've already differentiated, they don't need any help to go kill things. Memory B cells require Th2 help to secrete antibody. That is not true. Memory B cells do not need to present antigen in order to differentiate into plasma cells. They've already encountered antigen, and now they're memory cells. They're just waiting around to encounter it again. Dendritic cells are the only antigen-presenting cells capable of activating naive T cells. That one is true. At least in our version. It's probably true that macrophages can do it to like a really small extent, but dendritic cells are really the only cells that you need in order to do it. Macrophages alone wouldn't be sufficient. Which is true for IL-2. Enhances killing by macrophages. I'm actually going to give you the answer to this one. IL-2 is a growth factor for all lymphocytes. And that's important for T cell proliferation and natural killer cell proliferation. So IL-2 is a growth factor for lymphocytes. It's made by Th1 cells. Which of the following is a complement component involved in the formation of the alternative pathway C3 convertase? Factor B, factor I, factor D. Well, factor B is certainly involved. It binds to C3b, and then it gets cleaved by factor D into B flat, right? Into big B, little b. So this one's going to be A and C. Factor I is a breakdown protein. It breaks down, it cleaves C3b into inactive components. So it's involved in breakdown of complement and regulation of complement, not in formation of the convertase. Which adhesion molecules allow neutrophils to adhere to the vascular endothelium? LFA1, that one is certainly involved, right? It binds to ICAM1 and 2. P-selectin is involved. That one's expressed on the endothelium, and it binds to the sialyl Lewis X blood group on neutrophils. CR3 is also involved, can only bind to ICAM1. So this is all the above. Which is true for natural killer cells? Probably one of my favorite cells. Multiple answer. They can induce apoptosis in target cells. Yep, that's how they kill. If a virus-infected cell manages to avoid targeting by natural killer cells, it's more likely to be killed by CTL. That one is true as well. Remember that the way that viruses can avoid the host cell being killed is by downregulating MHC1. If they downregulate MHC1, they're going to be killed by natural killer cells. However, if they don't downregulate MHC1, then they're going to be targeted by CTLs. CTLs are specific for MHC1, okay? So they're kind of screwed either way. These two cells work together to make sure that target cells are killed regardless of MHC1 expression. One trigger for natural killer cell killing is fixed IgE. That one is not true. Natural killer cells don't respond to IgE. Basophils and mast cells respond to IgE. They are considered a type of innate lymphoid cell. That one is true. They are one of the classes of ILCs. Cytotoxic activity of natural killer cells is amplified by adaptive immunity. That one is true. Think tumor necrosis factor and IL-2 for that, made by Th1 cells, right? They are an important cell type involved in macrophage activation. This one is true. Natural killer cells make interferon gamma, which is one of the best ways to activate a macrophage. It makes them ruthless killers. They are a major target cell for type 1 interferons. That one is also true. So type 1 interferons are going to bind to receptors on natural killer cells and make them into really efficient killers. It makes them anywhere from 100 to 1,000 times better at killing. They are inhibited by the presence of MHC2 on the target cell surface. That one is false. So it's not MHC2 that they're inhibited by, it is MHC1 that they are inhibited by. Okay. Which of the following is a mismatch between enzyme and substrate? MASP2 and C2. Nope, that one is true. MASP2 cleaves C2 in your lectin pathway. C3b, B flat 3b, and C3. This is your alternative pathway C5 convertase. So that one's going to be a mismatch. Let's look at these other ones though. Here we have the classical lectin pathway C5 convertase, so that one is not a mismatch. Factor D and factor B, that one is not a mismatch. Factor D cleaves factor B. And then here we have the alternative tickover pathway C3 convertase. So that one's not a mismatch. Factor B and C3b. Nope, that one is involved down of C3b. So the answer is B. Which of the following is incorrect regarding adhesion? IL-8 enables LFA to firmly link to neutrophil cytoskeleton via talon. That one's true. So that one's not incorrect. PCAM will bind to PCAM on a neighboring cell. That one is true as well. PCAM is a ligand for itself. Rolling adhesion is mediated by selectins binding to Sila-Lewis blood groups on leukocytes. That one is true as well. ICAM1 is constitutively expressed, whereas ICAM2 is induced by inflammation. That one is false. ICAM1 is induced by inflammation only, whereas ICAM2 is always expressed on the endothelium. Okay, so don't mix those up. I doubt Sanders would even ask about that distinction, but just in case, it's part of my practice exam. What specific class of antibody may serve as a B cell receptor on a naive B2 B cell? Notice it's naive, okay? All classes of antibody can serve as a B cell receptor, but when it's a naive B cell, they express only IgM. These are B cells that have not encountered antigen before. They've not received T cell help. They've not presented antigen. They have IgM. They also have IgD, but that's not really as important as the fact that they have IgM. Which of the following adhesion molecules promotes initial and transient binding to vascular endothelial cells? Integrins, nope. Those promote tight binding. Selectins, yes. So that's probably going to be our answer, right? IL-8, nope. That one promotes the linkage of integrins to the actin cytoskeleton, which is involved in tight binding. So nope. Which is true for B lymphocytes? Antibody secretion, somatic hypermutation, and class switch are all dependent on T cell help. That one's false. Antibody secretion does not require T cell help for any B cell. For B2 B cells, they can make IgM without T cell help. So they can secrete IgM without T cell help. However, class switch and somatic hypermutation, those are dependent on T cell help. Okay, so if this little piece wasn't there, this one would be true. B lymphocytes is activated by MHC2 proteins expressed by antigen presenting cells. That one is false. B cells do not respond to MHC2. T helper cells, CD4 positive T cells, they respond to MHC2. Okay? B cells actually express MHC2, and they use it to present antigen to T helper cells. Can produce cytokines. That one is 150,000% true. T cells are actually a pretty important source of cytokines, something that's kind of overlooked a lot of the time. Deficiencies in which of the following would predispose an individual to parasitic infection? IL-4, yep. IL-4 is involved in the IgE response, and IgE is antiparasitic. Cytotoxic T cells, nope. Those are involved in defense against viruses and intracellular bacteria. Those are your CD8 positive T cells when they are effector cells. Eosinophils, absolutely. Eosinophils are one of the most important cell types in fighting off parasites. So this one's A and C. Okay. This is what the exams have historically looked like. So hopefully this is helpful to your exam tomorrow. Which of the following is anti-inflammatory? Transforming growth factor beta, yes, that is an anti-inflammatory cytokine. It's going to tell T cells to shut up and stop whining. Treg cells, yes, they are the source of transforming growth factor beta. They are also a source of IL-10. And IL-10 is also anti-inflammatory. It's going to suppress macrophage inflammatory cytokine production. So all the above for this one. Which is true for innate immunity? May involve IL-1, yes. May involve innate lymphoid cells, yes. And may involve tumor necrosis factor, yes. So this one's all of the above. Which is true for class switch? Requires T helper cell activation. That one's true. You're going to need T cell cytokines to undergo class switch. B cells must first bind antigen and present it to CD8 positive T cells. That is false. They don't present antigen to CD8 positive T cells to receive, class switch inducing cytokines. They present antigen to CD4 positive T cells, your T helper cells. Okay, so make sure you read carefully on the exam. Read carefully, because if you hadn't read the 8 there, you would have probably said that was true. Genetic rearrangements involve the VDJ region. That one is false. Okay, you're not changing the VDJ region. The VDJ region is the part of the genome that encodes the antigen binding site. In class switch, the antigen binding site doesn't change. Rather, the constant region changes on the antibody. It becomes, different. So you don't change the VDJ region, you change the C region, the constant region. Okay. Which of the following cytokines are associated with increased vascular permeability? IL-10, no. Tumor necrosis factor, yes. And none of these other ones. So tumor necrosis factor is our answer here. IL-4, nope. IL-17, no. Which of the following is a soluble cytoplasmic receptor that can cause innate immune cell activation? So notice soluble. NOD2, that one is soluble. Hey. That one's in the cytoplasm, and it detects peptidoglycan components. Dectin-1, that one's not soluble. It's on the cell surface. Toll-like receptor is also on the cell surface. So NOD2 is your answer here because it's the only one that's soluble. It's cytoplasmic. Which of the following cells would recognize antigen in the context of MHC class II molecules? This one's going to be your CD4 positive T cells. B cells don't respond to MHC2. CD8 positive T cells do not respond to MHC2. They respond to MHC1. B cells can respond to pretty much any kind of antigen. Okay, it doesn't have to be an MHC molecule. And in fact, they will not respond to an MHC molecule. Pretty much ever. Which of the following can directly serve as an opsonin, meaning marks things for phagocytosis? IC3B, yes. IC3B is an opsonin. It is a ligand for multiple different complement receptors, including CR1 and CR3. So it is an opsonin. C-reactive protein, yes, on its own is an opsonin. Its receptor is the Fc gamma R1 receptor and Fc gamma R2A. So it actually uses IgG receptors in order to opsonize things, weirdly enough. IgG, obviously that one is an opsonin. It's going to mark things for phagocytosis. So all the above for this one. Which of the following can be an effect of interleukin-1 beta? Just think of this as interleukin-1, IL-1. Fever, yes. The cytokine can diffuse through the bloodstream and act on the hypothalamus, causing production of prostaglandin E2, which binds to muscle cells and fat cells and causes the mobilization of stored energy and burning of calories with no real ATP generation. And that is going to increase the temperature set point of the body and cause fever. So this one acts on the brain to make fever happen. LFA1 expression on endothelial cells. No. LFA1 is not expressed on endothelial cells. LFA1 is expressed on neutrophils and monocytes and T cells. Release of Weibel Palade bodies containing P-selectin? Yes. So A and C for this one. Remember those are stored granules inside endothelial cells. I talked about those today. Which of the following can activate the alternative pathway of complement? Fixed IgA, that one certainly can, right? That's one of the ways that this becomes activated. C-reactive protein, no. C-reactive protein is a classical pathway activator, not alternative. IC3B, no. IC3B cannot activate the alternative pathway. Only C3b can activate the alternative pathway. IC3b, however, is still an opsonin, okay? So you don't get a positive feedback loop with IC3b, you don't get alternative pathway activation, but you do get opsonization still. Okay? So only fixed IgA for this one. Which of the following are transcription factors in the immune system? NF-kappa B, yes. That one turns on inflammatory cytokine genes and anti-apoptotic factors, and it transcribes enzymes involved in reactive oxygen and nitrogen species production, right? It turns on inducible nitric oxide synthase, for example. RAC2, this is not a transcription factor. RAC2 is a G protein, and it turns on, it assembles NADPH oxidase. That is its job. IRF3, yes. IRF3 is it's one of the transcription factors for the type 1 interferon cytokine genes. So it's going to cause the cell to make interferon alpha and beta. So A and C for this one. Which of the following subclasses of antibody is most closely associated with allergic responses? You should just know that this is IgE. That's something that you should know. You need to know your antibody classes, okay? So SI Slideshow 4 is going to help you out with that one. Which activates PREX1, the guanine exchange factor for RAC2? This one is G beta gamma. Okay, G beta gamma. We didn't cover this until today, but G beta gamma will activate PREX1, which then activates RAC2, which then assembles NADPH oxidase. Remember G alpha, it turns on phospholipase C, which cleaves PIP2 into DAG and IP3. Which of the following substances will increase natural killer cell killing? IL-12 is not going to do it. No, oh, I'm sorry. IL-12 is going to do it. My mistake. IL-12 has two important activities, right? Its first important activity is that it activates natural killer cells to become better killers. Its second important activity is that it causes it causes Th1 differentiation. Okay, so it promotes the Th1 response. So IL-12 is going to do it. Interferon gamma does not act on natural killer cells. Natural killer cells actually make interferon gamma, and it doesn't act on them. IL-6, so far as I know, does not act on natural killer cells, at least not in an important way. So the answer to this one is A, IL-12. Okay. IL-6, its job is to diffuse to the liver and cause the production of acute phase proteins like C-reactive protein, transferrin, lactoferrin, vitamin B12 binding protein, and etc. And IL-6 also promotes antibody production, and it can induce T-cell differentiation into Th17 cells and Treg cells when we're talking about that whole thing. Which of the following can provide a danger signal or co-stimulation to T helper cells? B7.1 or CD80. That one can certainly provide co-stimulation, right? That's the classic example. PAMPs binding to pattern recognition receptors like dectin-1 on a T cell. No, T cells are not stimulated by actual PAMPs. So they're not stimulated by microbes themselves. They're stimulated by antigen-presenting cell molecules, okay? So that one's not going to do it. IL-1, that one will do it. That one can provide a danger signal or co-stimulation to T helper cells. So this one's A and C, okay? So PRRs are not a way that T cells become activated. T cells are activated by MHC proteins and other proteins on antigen-presenting cell surfaces, okay, as well as cytokines. Which of the following cells express MHC1? This one's easy if you know the rule. MHC1 is expressed by all nucleated cells and platelets. MHC1 is expressed by all nucleated cells and platelets. What this means is that essentially only red blood cells do not express MHC1. So all of these except for the red blood cells express MHC1, okay? MHC1 is expressed by all nucleated cells and platelets, meaning only red blood cells are not expressing. Red blood cells are also called erythrocytes, if you didn't know. So hepatocytes, neutrophils, doesn't matter. All of these are going to express it. Which mediator cleaves into IP3 and DAG? Protein kinase C is definitely not going to do that. It's going to phosphorylate proteins to bring about reactive oxygen species production or degranulation. So it doesn't do that. RAC2, that one doesn't do it. RAC2 assembles NADPH oxidase. Phospholipase A2. Well, you think, well, it's a phospholipase. Maybe it cleaves PIP2. PIP2 is a membrane lipid. The answer is no. Phospholipase A2 cleaves arachidonic acid from the membrane. How are you going to remember that? It's a phospholipase. It cleaves membrane lipids that start with the letter A. Arachidonic acid starts with an A, right? Phospholipase A2 cleaves arachidonic acid from the membrane lipids. PREX1, nope. That one is the guanine exchange factor for RAC2. Phospholipase C, there's your answer. Phospholipase C. Phospholipase C is activated by G-alpha when it's bound to GTP. Okay, and it cleaves PIP2 into IP3 and DAG in the PIP2 pathway. Cationic peptides, 12 to 80 amino acids long, comprised of positively charged region, spacer region, and hydrophobic region. Produces proproteins, no disulfide bonds, but containing alpha helices. Which of these would it be? This one was actually an extra credit question. So if that makes you feel any better about not knowing this one, so be it. Lysozyme ain't gonna be it. Lysozyme is an enzyme that cleaves peptidoglycan. Histatins ain't gonna be it. Histatins are they're histidine-rich proteins, and I don't think they have alpha helices, and I do think they have disulfide bonds. Cathelicidins, they're not going to be it either. Difference is cathelicidins have disulfide bonds. The defensins have no disulfide bonds. Okay, and the lectins, these are carbohydrate binding proteins. They have a similar structure, but the important thing about them is that they bind to carbohydrates. Their structure is not really as important, and it's more variable anyway. Okay, at least I'm 92% sure that that one is defensins. So I'm not even sure. Neutrophils express which of the following? ICAM2? Nope. Intercellular adhesion molecule is expressed on endothelium. It's expressed somewhere where you have cells close together, right? Blood, cells are more spaced out than the vascular endothelial cells. They're directly linked to one another. So that is not the answer. Fc epsilon R1, they don't express the high affinity IgE receptor. Okay, this is expressed only on mast cells and basophils to respond to IgE. PCAM, they do express. Okay, PCAM they do express. CD31, that one's also expressed on the endothelium. That's the only one that's expressed on both leukocytes and the endothelium. Okay. Which of the following subclasses of antibody is the most concentrated isotype on mucosal surfaces? Mucosal surfaces, that is IgA. Mucosal surfaces, the class is IgA. You should just know that. IgA is mucosal surfaces and alternative pathway. IgG, that one's in the blood and classical pathway. IgM, that one is also in the blood and it does the classical pathway and it's a pentamer. IgE, that one is on mast cells and basophils and it binds to parasites and allergens. IgE causes allergies. IgD isn't as important as the other ones, but it's on naive B cells and it doesn't get secreted, so it really doesn't have, a function in defense. Which of the following substances will directly suppress a Th2 response? Notice the wording here, directly suppress. IL-12 does not directly suppress a Th2 response. However, IL-12 will promote a Th1 response, and those Th1 cells are likely to make other cytokines that will directly suppress the Th2 response. So this one, you might say, indirectly suppresses the Th2 response, but does not directly suppress Th2. IL-1, nope. That one actually is a general activator of T helper cells. Interferon gamma, ding ding, that is the answer. Interferon gamma made by Th1 cells, it's gonna inhibit the proliferation of Th2 cells. All right, so it's gonna downregulate or suppress the Th2 response. Which of the following cytokines promote a Th2 response? IL-12 and interferon gamma both promote Th1 responses. IL-9 is definitely going to promote Th2 response. B cells with high levels of MHC2, foreign peptide complexes on their cell surface also express which? So this means that the B cell has encountered antigen, it has taken up the antigen, it has shredded the antigen into a bunch of bite-sized pieces, and it has loaded those pieces into it has loaded those peptides into MHC2. Now it's trying to present MHC2 to a T helper cell. So the B cell is looking to receive cytokines. And B cells are affected by almost every cytokine. So it's going to be all of the cytokine receptors. I had one on here, I had another answer choice on here in the old version that I removed. So heads up on that. I don't think it's fair to ask you about ICOS ligand or anything like that. So I took it off. Which of the following can cleave and activate C2? C1q, nope, that is the one that binds to IgG, IgM, and C-reactive protein to kick off the classical pathway. But it's not an enzyme. It's a big, long, stalk-like protein with a flower head at the top. C3b, B-flat, nope, that one is the alternative pathway C3 convertase. MASP2, yep, MASP2 is an enzyme that can cleave and activate C2. That is in the lectin pathway. MASP1, that one doesn't, or actually it does cleave C2. It cleaves about 40% of the C2. And that's actually something I forgot to mention, I think, when we went over complement. Sanders talked about it last year, but MASP1 is a promiscuous protease, right? So it can cleave C3, it can cleave C2, and it also cleaves and activates MASP2 during the classical path, or excuse me, the lectin pathway. So MASP1 is a promiscuous protease, and its ligands include MASP2, C3, and C2. So apologies for not mentioning that. Then we have C1s, and that is one of the enzymes that can cleave and activate C2 indeed. Remember, in the classical pathway, it's in alphabetical order for the C1 proteins. C1q binds, then C1r is activated, it cleaves C1s. C1s is the active enzyme that cleaves C4 and C2. So QRS. C-reactive protein, nope. C1r, no. C4b, no. Which of the following suppresses the Th1 response? That would be IL-4, not interferon gamma. That one promotes the Th1 response. And IL-10. So A and C on this one. Which of the following are true for C-reactive protein? May be induced by IL-6, acting over. That's true. May serve as an opsonin on its own. That one is also true. So it can bind to pathogen surfaces, and it can mark them for phagocytosis by two different IgG receptors. May activate complement by the classical pathway. That one's also true. Right. Naive T helper cells express which of the following? IL-2 receptor. That is true. They express only the moderate affinity IL-2 receptor, which we'll learn more about on next exam, but they do express a receptor for IL-2. All lymphocytes express a receptor for IL-2. CD8, nope, that one is expressed by cytotoxic T cells. CD28, yes, that is the receptor for B7 on the antigen presenting cell. So this one's going to be A and C. Which of the following cells under normal conditions are primarily found outside the circulatory system? Basophils, those are mainly in the blood. Okay. On the other hand, mast cells are in tissues, right? Monocytes are also mainly in the blood. However, B cells, they accumulate in lymph nodes. They accumulate in the follicles and lymph nodes, right? So they are mainly outside the circulatory system. Okay. B cells is the answer to that one. Which of the following is true for the acute inflammatory response? Monocytes are the second phagocyte from the circulatory system to appear in the tissue at the site of trauma. That is true. Neutrophils are the first phagocyte to appear. Monocytes come a little bit after neutrophils. And then T cells come a little bit after monocytes usually. Okay, so that one's true. Neutrophils function as phagocytes and also present antigen to T helper cells. That one's false. They do not present antigen to T helper cells. They don't express MHC2. One of the early vascular changes is increased vascular permeability. That's obviously true. Histamine, tumor necrosis factor, all of those things, leukotriene, B4, all those things increase vascular permeability. Which are amphipathic peptides secreted in salivary glands that kill fungi and gram-positive bacteria and may quicken oral wound healing? So they're made in salivary glands. That would be the histatins. Okay, histidine-rich peptides made in the saliva. Which generates the superoxide anion? You learned that one today. That would be NADPH oxidase. And then that converted by superoxide dismutase into peroxide, hydrogen peroxide. Hydrogen peroxide can generate hypochlorite by way of myeloperoxidase, or it can be broken down into water and oxygen by catalase, which is expressed on pathogens. Which can serve as the B cell receptor? Multiple answer that might have given it away. That would be all of these. B cells in general can have any class of antibody as their B cell receptor. However, naive B cells only have IgM and IgD as their classes of antibody that are their B cell receptor. I will say that a single B cell is only going to be making one class of antibody unless it's expressing IgM and IgD. Those are expressed together. So it's only going to express IgG if it's expressing IgG, or the same goes for the other two. But they can all serve as a B cell receptor. Which of the following would most likely elicit a thymus-dependent response? When you see thymus-dependent, think antigen presentation to T cells. Okay, so you think protein. Proteins are thymus-dependent responses. So a surface-exposed polypeptide receptor molecule. That one is a possibility, right? It's protein, so it can be presented to a T cell. Polysaccharide capsule. Nope, that one is not protein. It's made of polysaccharide or sugar, whatever you want to call it. A phospholipid found in a cytoplasmic membrane. Nope. So this one's going to be A. Now we're going to learn more about this weird thing called CD1 on our next exam. And this one's going to look a lot more plausible. But CD1 generally doesn't just present normal phospholipids. It presents these long lipids that are like 40 carbons long. Anyway, which of the following cells would respond to MHC1 peptide complexes? That's going to be your CD8 positive T cells. None of the other cells respond to MHC1 loaded with peptides. So cytotoxic T cells. Which of the following cannot be a result of T cell help to a B cell? Class switch to IgG, M, A, or E, or none of the above. This one's going to be class switch to IgM. Remember that B cells always start with IgM. They always start with IgM. It is the earliest class. They don't even need T cell help to secrete IgM. And once they switch to another class, they cannot switch back to IgM. Okay, switches are permanent. However, they can switch to a different class. So you can switch to IgA and then IgG and then IgE, for example. I think IgE is actually earlier than IgA. Anyway, they cannot class switch to IgM. They start with it. So, and they can't switch back to it either once they've switched to something else. Which of these can dislodge bound B flat or big B little b? Decay accelerating factor can certainly do that. That's why it's called decay accelerating factor, DAF. CD59 cannot do that. CD59 is a protein on host cells that binds to complement component C8 and prevents C9 from polymerizing on the host cell and forming a membrane attack complex. CR1 can indeed dislodge B flat though. It's one of the many things CR1 is amazingly capable of doing. So this is A and C. Now, which of the following is false regarding complement? This is one of my hard ones, I think. The C5a receptor can sensitize phagocytosis by macrophages. That is true. C5a receptor is a G protein coupled receptor. It triggers the PIP2 pathway, and it makes macrophages better at phagocytizing things. The classical pathway can initiate the alternative pathway, but the alternative pathway cannot initiate the classical pathway. This one is not false. This one is true. So the classical pathway can produce C3b, which can bind to the pathogen surface and trigger the alternative pathway, right? So the first part, that's true. The alternative pathway, however, cannot initiate the classical pathway because it doesn't make any intermediates or mediators that trigger the classical pathway. The classical pathway is triggered by IgG, IgM, or C-reactive protein. And nothing about the alternative pathway suggests that you would get any of those things attaching to the pathogen in the alternative pathway. C3b has an active site that may be capable of cleaving C5. This one is not true. This one is your false answer choice. C3b is not an active enzyme, okay? In the classical and lectin pathways, C2b is your active enzyme. And in the alternative pathway and the alternative tickover pathway, B flat is your active enzyme. Okay, C3b is never an active enzyme. It's not an enzyme at all. It just binds to things and changes them and marks them for phagocytosis. Red blood cells are involved in the major mechanism of how complement is cleared from the body. That is not false. So that's called immune adherence, and they use the CR1 to do that. There's a slideshow in the complement, or there's a slide in the complement slideshow on that. Factor P properidin positively regulates complement. That's true. Factor P is going to bind to alternative C3 convertases and protect them from degradation and make them last longer. C9 cannot cause membrane attack without C8 being present. That one is also true. C9 will never polymerize unless C8 is already present in the membrane. So the answer here is C. Okay, that one is false. Okay, and last one. Which of the following components is mismatched to its function? Diacylglycerol activation of phospholipase A2. That one is true. That's one of the things that diacylglycerol does. IP3, release of calcium ions from the ER. That one is also true. It binds to its receptor on the ER, causing it to open. Serves as a channel to leak calcium ions out. PLC, generation of arachidonic acid via cleavage of membrane lipids. That one is your mismatched choice. PLC does not cleave arachidonic acid. Phospholipase A2 is what cleaves arachidonic acid. PLC cleaves PIP2 into DAG and IP3. Okay. DAG, activation of protein kinase C. Yes, that's another thing that DAG does. So it activates protein kinase C, and it activates phospholipase A2. Phospholipase A2 cleaves arachidonic acid from the membrane. Calcium, activation of proteins that bring about cytoskeletal rearrangements. That one is not mismatched. Calcium does indeed bind to calmodulin, which then brings about cytoskeletal rearrangements, chemotaxis, and all that fun stuff. So, that is the practice exam. Realize that this is probably harder than the actual lecture exam for good reason. Because, you know, I'd rather bring a gun to a knife fight than a knife to a gun fight.